Hey everybody, happy Thursday, Friday. Just here to give you a lesson on the spectrum of dominance while I'm out. So thanks for being flexible with that. So far in genetics, we've been talking about examples of monohybrid crosses. And we start with that because they're just the most straightforward type of crosses because you're just looking at a single character, right? It might be flower color, it might be height, it might be eye color. And in all the examples we looked at so far, we've seen an example of what's called complete dominance. What that means is that you have two characters, right? Or sorry, two traits within a character. So like purple flowers and white flowers. And in complete dominance, what this means is that one of these traits, in this example, purple, completely dominates over the white. And today what we're going to do is take a closer look at why there's dominance. How does dominance actually work? Okay, because in the freshman level version, and maybe even in Mendel's time, the idea of like why dominance happened was more about like one being stronger or more powerful than the other. And in reality, it's not really about that. Really, the truth of it is it's about a difference in how the cell follows genetic instructions and what those instructions, those genes actually code for, and what protein is made and how much protein is made on the phenotypic side of things. So that's what we're going to look at today is, what is dominance all about? And then we're going to look at some exceptions of dominance. We kind of call these exceptions to Mendel. And sorry, these are examples of dominance that are not the same as complete dominance. It's a little bit more blurry. And what I hope you get out of it, and I'll point to this at the very end when I give you an example of Tay Sachs disease, is that in reality, genetics is not really that simple. Okay, we want it to be simple. We want it to be as simple as brown eyes and blue eyes. We want to be simple as calculating 3-to-1 ratios. But in reality, genetics is a really, really, really complicated field. And it's because our genes and the way that our cells use our genes is really complicated. So today I'm just going to give you a little glimpse of how complicated it is and look at some different case studies, and some of them you might be familiar with and some you might not. And so to do this, I'm going to actually be using the whiteboard for most of this because, and I know once I click on this, you're not going to see me, but that's fine. I actually prefer that. It's less pressure on me. But I'm going to be using the whiteboard. As we go through this, I'm going to give you some notes and draw out some examples and stuff. And then there will be points in today's lecture where I'm going to ask you to pause the video and work on a practice problem. All right. So when you hear me say that, go ahead and pause it. I'll give it a few seconds for that pause, and then when I come back, I'll walk you through how to do it. But please take advantage of those opportunities to try it yourself first before you see me actually do it. Okay, so today we're going to be talking about something called the spectrum of dominance. And the spectrum of dominance refers to a kind of a sliding scale, okay, of different types of dominance. The word spectrum just, literally means it's a sliding scale, right? If you think about the color spectrum, right, it's not very clearly, like, it doesn't just go like straight from red to orange to yellow to green to blue. It kind of blends its way across, and that's why we call that a spectrum. We also use the word spectrum to describe gender and sexuality, because the more we learn, the more we realize that it's not biologically even. It's not just as simple as male and female. There's a lot in between in terms of biological anatomical structures, but also what does it mean to have gender? And so we use the spectrum a lot to describe situations where it's not really clear cut one thing or the other. It could be a blend of different things, right? And the way I want you to visualize this today is we're going to be looking at really three aspects or three ends of this dominance. Okay, on one end over here, we're going to have, as I mentioned, something called complete dominance. All right, and as you would expect here in complete dominance, sorry, that line's not perfectly flat. In complete dominance, you have one trait that's going to completely dominate over another. And actually, that's kind of where I want to start here today. Okay, is with this particular slide, we're just going to look at complete dominance. In complete dominance, the example that I'm going to use is the one that we've been talking about. We're going to see that purple flowers are going to be fully dominant over, let me change colors here, white flowers. Okay, and what does that mean? How does that actually play out in terms of the relationship between genotype and phenotype? Well, let's take a look at that, right? In the genotypes here, there are three possible genotypes we could have had, right? You could have homozygous dominant, big P, big P. You had heterozygous, big P, little p, and then homozygous recessive. Okay, in a monohybrid cross, these are the only 3 genotypes you could have. And in complete dominance, what's really interesting here is, it's no surprise when we look at it phenotypically, right? When you are homozygous dominant, you're going to be a purple flower. If you're a homozygous, sorry, that's an ugly flower there. When you're a homozygous recessive, you're going to have a white flower. What was kind of the interesting one to Mendel, and it's interesting to us, I don't know if you find it interesting, is that the heterozygous ends up having the same purple flowers as the homozygous dominant. Okay, and to understand why this actually happens, we need to reframe the way we think about the coloration of flowers. Okay, and because here's what actually happens when the flower is growing, right, and it produces these petals. What you have to imagine is kind of think about this like paint on a piece of paper or dye in a fabric, right? If fabric doesn't have dye on it, or a white piece of paper doesn't have any paint on it, what color is it by default? It's white, right? And so what's going to happen here is, let's say that this homozygous dominant plant gets 2 copies of the purple allele. What that means then is that the plant is going to make 100% of the pigment needed to color the flowers purple. Okay, and here's kind of how we think about it. Let's say arbitrarily that it takes 100 units, okay, of purple pigment to make this whole flower purple, right? What we're saying here is that if you have 2 copies of the big P dominant gene, that's going to mean that you're going to make all 100 units, and that's going to lead to a flower that is all completely purple. Okay, which I can shade in like this. Okay, now let's back up a little bit. If, on the other hand, if you have little p, okay, I know it's kind of hard to see because I'll put a line under it to show it's lowercase. Does that mean that this flower has a gene that codes for white pigment? The answer is actually no, okay, because there's actually no such thing as white pigment as far as I know. Okay, what we have is if something lacks pigment, then it's just going to be white, right? And so if you think about it this way, it's not about purple proteins versus white proteins. What it is it's a difference between purple proteins and no proteins. And that's actually why white flowers have white color. It's because how many of those units of purple are they making? They're making 0, right? And the result of that is then a flower that has no color to it. Okay, so the shift in the way our thinking here is it's not that you have 2 competing colors, right? Another way to reframe this, and I would write this down, is that, and if you need to pause at any point, this is the nice thing about screencasts, you can just pause it and take down the notes. White is not its own standalone color. White is a lack of purple, okay? And that is actually why we see these 2 characters, right? Now, again, the interesting thing is what happens in the heterozygote, right? You have this flower now, in all of its cells, one of the chromosomes says make purple, the other one says not white, but it says don't make purple, right? And what does the cell do? How does it respond? Well, as it turns out, in the heterozygotes, having 1 copy of the dominant allele is enough to trigger that flower to produce the same amount of pigment as an individual that is homozygous dominant. Okay. In other words, the production of protein, the production of the purple pigment here is disproportional to the genetic information, right? The genetic information here, if you think about this in terms of votes, right? We got an election coming up next week. In the genetic vote in the heterozygote is 50% of it saying make purple protein, 50% of it is saying don't make purple protein, in other words, white. And yet the flower is going to make 100% of that purple protein, the same amount of protein as an individual who has 2 votes that are both saying make purple. Okay, and guys, that's what it comes down to when we talk about complete dominance. Okay, what we're saying here is this, and I'll go ahead and type this sentence out for you, is that in complete dominance, just 1 copy of the dominant allele is enough to produce the same quantity of protein as, I gotta make some more room here for you. Same quantity of protein as an individual with 2 copies of the dominant allele. In other words, the homozygous dominant individual. Okay, so the person that the individual has one copy of the dominant allele, that's the heterozygote, right? And that's going to be an important thing that we're going to point out today. Okay, is that if you're trying to figure out what kind of dominance is being used, actually, let me just move this whole text over here. I don't need all that space. If you're trying to figure out what kind of dominance is being used here, simply look at the heterozygote. Okay, if the heterozygote looks the same as the homozygous dominant individual, then you know this is a case of complete dominance. Okay, now does this apply to other examples? And the answer is absolutely yes, right? Another example we saw in pea plants was the difference between tall and dwarf plants. What is the difference between tall and dwarf plants? Is it because there's a tall gene and a dwarf gene? No, there's no such thing as a dwarf gene. Okay, the tall gene, the height gene, actually codes for a growth hormone that tells the plant to grow, right? And so really what it is, it's not a difference between tall and dwarf. It's about the difference between a plant that's told to grow and a plant that's not told to grow, right? In other words, the recessive allele usually is some kind of mutation that prevents a certain protein from being made correctly. And as a result of that, the plant doesn't make the protein, whether it's a purple pigment or a growth hormone or even a certain enzyme. I don't know if you guys looked in the book, but there's an example of wrinkled peas versus smooth peas. And without getting too much into the detail here, the difference between them is that one of them makes a correct enzyme that then causes water to move into the, I think I might get these mixed up, but the water is going to either move in or out of the pea. And so some peas are going to be smooth because they have the enzyme, and some are going to be wrinkled because they don't have the enzyme. Okay, so a lot of times complete dominance is about a protein versus a lack of a protein. It's not really about one being better or stronger than the other. Okay, all right, let's move into the next one. Let's go to the middle now, and we're going to look at something. I'm going to just type this in, and I'm going to start a new slide for this. Something called incomplete dominance. Okay, and hopefully. There we go. Okay, I just wanted to squeeze that in right there. So in the middle, things start to get a little bit more blurry. Okay, and we have this thing called incomplete dominance. Let me make a new slide for this. In incomplete dominance, the amount of protein made is proportional. Sorry, I cannot type. Proportional to the genetic information. Okay, and so the example that we're going to look at here today is the example of Snapdragons. Okay, in Snapdragons, there are two alleles. Okay, red and white. Okay, similar to purple and white. The difference here, though, is that these two alleles don't exhibit complete dominance over each other. Instead, we have something called incomplete dominance, right? And to understand how this is set up, let's go ahead and set up our little chart here to keep track of things, okay? We're going to start by looking at the genotypes, okay? I'm going to track the percentage of the vote, okay, in terms of like what are the genes actually telling that cell to do in terms of the color being made. And then we're going to track how much actual protein is made. And then the result of that is what phenotype do we actually see. Okay, so we're going to write out the genotypes. Now, here's kind of the first big change. Okay, if we know something is using incomplete dominance, then what we're not going to do is use our normal system of capital and lowercase letters. Okay, we're not going to do that because if you write it only with big R's and little r's, the standard practice in genetics is that people will see that and they assume you're talking about what? Complete dominance. Okay, it assumes that red is completely dominant over white. Now I'm telling you right now that in Snapdragons, we're using incomplete dominance. And so we're going to write things a little differently. The way we're going to write it is we're going to use the letter C to represent color, right? And we're going to say that both of them are going to use the letter C, capital C, but above that, we're going to put R and W. Okay, so the CR stands for the red allele and CW stands for the white allele. All right, So we're not really using uppercase, lower cases here because, again, that's going to tell us that it's complete dominance. Okay, so let's go ahead and look at the options here. Let's start with the easy one. Okay, if a flower is CRCR, it's homozygous red. Okay, and you could say it's homozygous dominant, but again, it's kind of blurry because this is a spectrum, so it's not super clear. In this case, what is our genetic vote, right? If both of these represent one vote, each of them are telling the cell, my vote, my 50% of the vote is saying make red pigment, right? And both of them are saying that. So what that means is that in this individual, 100% of it is saying make red. Okay, let's make, there you go. All right, so 100% of the vote is saying go ahead and make red, all right? And if, let's say that the flower is kind of the same way as what we saw before, okay? You have the flower petals, they're made to be white, and then in order to make it completely red, it takes 100 units of red pigment to fill all that. What's going to happen here is, remember, the amount of protein made is proportional to the genetic information. And so if the genetic information is saying 100% says make red, then what percentage or how many units of protein is going to be made? We're going to go ahead and make 100% of that red protein, okay, or that red pigment. And when that red pigment is injected into the petals of the leaves, then what you end up with are flowers, and I'm going to erase that just so I can use that again later. It's going to produce a phenotype, which is our flowers are going to be red. Sorry, it's a little messier today because my mouse pad's on the bed. So it's a little squishy. All right, so no surprise there, right? That's no big deal. Let's go to the white side. Okay, same idea. If you have an individual that is homozygous for the white allele, what is your genetic vote? Is it that it's 100% white? No, it's not. Right? What it's saying is 100% of the vote is saying don't make red. Okay, remember, white is the absence of red. Okay, so the genetic vote here is basically saying what percentage of it is saying make red. The percentage here is going to be 0%. Okay, because the alleles that are sitting on that chromosome, remember, these are representative chromosomes, right? This chromosome, both these chromosomes have a gene where the genetic code has some kind of mutation that is saying don't make the red, or it's incapable of making red protein, right? And so if the vote is saying don't make red, then how much red protein are we going to make? We're going to make 0% of that. And if we don't make any protein, no pigment is injected into the petals, and it's going to remain white. Okay. Now, here's the fun one. What if we get one of each? Okay, if they're heterozygous, and remember I told you guys, if you want to know what kind of dominance it is, look at the heterozygote. So we have a heterozygote here that has a copy of the red allele and a copy of the white allele. What's going on with the vote here? Well, the vote is now split, right? Kind of like what we saw in the past. One allele is saying make red, and the other one, the other 50%, is saying don't make red. Or in other words, 0% make red. I don't know if that's confusing to you, but you can look at it either way, okay? So this side, one allele saying make red, the other side is saying don't. And here's, again, the important piece, okay? In incomplete dominance, the amount of protein you made is proportional to your genetic information. And so if only 50% of the genes are saying to make red, how much red protein is made? The answer is only 50% of it is red. Okay, and if you only make half the protein, here's the cool part. You still want to fill all this petal, right? But if you use half the amount of pigment and you spread it out in the same surface area, is it still going to be red? No. What it's going to be is a lighter version of red, which is pink. Okay, and so in Snapdragons, you have red flowers, you have white flowers, but then you also have pink flowers. And what people want to kind of often misunderstand, right, is because we think about this like paint, is that the way you get pink is to mix white paint with red paint, okay? But I want to point out that in this case, the flower is not pink because it's mixing red and white. It's getting pink because it only made half of the red and it's spreading it out in the same area, and so it appears to have a lighter color. So the pigment is exactly the same, but it's just in lower concentrations, and therefore it produces pink. Okay, so this brings us to our first little practice question for today. Okay, if I do that, I'm gonna go ahead and write this out, and then we'll solve it really quick. Okay, if you mate a red flower with a white flower, what are the ratios? Okay, the phenotypic ratios. I'm talking about the color here. What are the phenotypic ratios in not the F1, but the F2 generation? Okay, and if you're confused by that, what you need to do is you got to mate the red and white flower together first to get the F1, and then you take two of the F1s and mate them together to get the F2. Okay, so go ahead and pause this YouTube video and see if you can write out the parent genotypes, do the crosses, get some Punnett squares going, and then tell me what are the phenotypic ratios in the F2 generation. All right, I'll see you guys in a few minutes. Okay, hopefully you guys actually paused it. Otherwise, this is going to be really awkward. Hopefully you got a chance to work on that problem, and let's take a look at how this works. Okay, if we have a red flower, and I'll draw this out so we have a visual here. I don't want pink yet. There we go. Okay, so if we have a red flower, we know that this is CR, right? And we're going to cross that with a white flower, which we know is CW. Just by looking at this, we don't even need a Punnett square. We know 100% of the offspring are going to be CW, which means what color are they going to be? All the F1s are going to be pink, right? So this is a P generation. Here's your F1 generation. All of them are going to be pink. To get the F2 generation, what we need to do is mate two of these together. So two pink flowers. Okay, let me try to be consistent here. CW. Right? And this is where we want to do our Punnett square, right? We've done this with monohybrid crosses before. We have to ask ourselves, what are the possible gametes that each of these parents can make? And so hopefully what you remembered is that homologous chromosomes get separated during meiosis. So this parent can either give a CR or a CW, and this one, same thing, can only give a CR or a CW, whether it's sperm or egg, right? And so what we're going to end up doing is making a Punnett square. That's going to look, let me draw my Punnett square here. It's going to look something like this. It's going to be 2 by 2 because I have 2 possible sperm, 2 possible eggs, just like we've done in the past. And let's go ahead and put them in, okay? The sperm either has CR or CW. And the egg is, again, going to be CR or CW, right? And so what does that mean for the offspring? Well, let's go ahead and put it together. Both of these are going to be CR. This individual is going to be what color, guys? Red, right? CR plus CW, that's going to make a heterozygous individual. Again, I apologize for the messiness. Turns out the bed does not make a great mouse pad. And then this one also is going to be CRCW, which is, again, going to be pink. But this last one is going to be homozygous for the white. And so you're going to end up getting, what's really interesting, is not a 3-to-1 ratio, but a 1 to 2 to 1 ratio, where 1 out of 4 is red, 2 out of 4 is pink, and 1 out of 4 is white. Okay, and that, again, this is what it means to have incomplete dominance. Okay, and I want to emphasize that pink is not a blend of red and white. What it represents is a intermediate, right, of red and white. It's a halfway point between making no pigments and making all the pigments. That's why we get the pink. Okay, so that's incomplete dominance. I can hear Colin crying out there. All right, this brings us to our last one. And actually, let me go back to our chart here. Okay, so let's add in something here. So we have an incomplete dominance. We had red flowers if you're homozygous. We had white flowers if you're homozygous. And then if you're heterozygous, it was a pink flower. Okay, so we'll draw that in there. And I mean, there's kind of a little dominance there, but it's a little bit blurry, right? And that's kind of the whole point. It's not black and white, like literally, it's not black and white. There's a little gray area there. Okay. Speaking of gray, there's actually, I think, was it in rabbits or mice? I can't remember, but in some kind of rodent, right? If it's a mouse or a rabbit, it's kind of a similar idea. Like black rabbits and white rabbits make gray rabbits. okay? And their hair, every individual hair is actually gray because gray is an intermediate between black and white, okay? So we have complete dominance, we have incomplete dominance. Now on the other side, okay, I know that that was not perfectly in the middle, but it's meant to be. On the other side here, we have their last example for today, and this is called co-dominance. Okay, if you can go back to your notes and add that in at the end of the spectrum, you have co-dominance. And in co-dominance, what we're going to see is two alleles that are equal to each other, right? The word co means that you're equal if you're partners, right? If you're a co-president of your club, that means one president is not higher than the other. You're equal to each other, okay? And as you would expect, the situation we're going to find here is that you're going to have two alleles that are actually equal in strength to each other. In other words, this is different than both examples before because remember, purple and white, right? Sorry, let me rephrase this. White is a lack of purple, right? White is a lack of red. Pink is an in-between between red and purple. But in this case here, A and B are actually equal to each other because A and B actually code for different proteins, okay? Not a lack of proteins. Now, I don't know if it's jumped out at you yet, but what example we're going to look at next. But if you're talking about A and B, we are definitely talking about blood typing, right? And so blood typing is an example of co-dominance. It's actually a little bit more complicated than that. And in co-dominance, what happens here is, let me stretch out my box here. In co-dominance, two alleles actually code for two different proteins, okay? And they are equal in strength or dominance to one another. Okay. Now, before I get to blood typing, let me give you a simpler example, just because I think we have enough time. I don't actually know how long I've been recording. I don't have a timer on here. So an example of this I think it's the Himalayan rabbits, actually, now that I think of it. What happens here is if you get a rabbit that is black, okay, let me make this a little thicker here. So if you have a rabbit, here's my rabbit with the ears, okay, that's black. And then you have a rabbit that is white, and you mate them together. What you end up getting is a gray rabbit, okay? And this one's a little bit tricky to understand here, but what happens here is that each hair kind of follows a different set of instructions, okay? Like the white, which codes for a lack of, is a lack of black, is actually followed as a full set of instructions, okay? And so if you were to zoom in on this gray rabbit, if this is an example of codominance, okay, instead of seeing every hair be gray, what you would find is that some hairs were white and some hairs were actually black. Okay, so white and black are both expressed simultaneously next to each other. Okay, in contrast to the mouse thing I was telling you earlier about, where black and white mix and the not mix, but they, there's a proportional production of the protein, and every hair is actually gray. Okay, that's kind of the difference. Okay, not the best example, which is why I don't really want to use this one as the main example, but that's a light example. A better example of codominance is in our blood typing. Okay, blood type is determined by these proteins. Actually, they're technically carbohydrates. Okay, but for purposes of our class, we'll call them proteins. They're called immunoglobulins. Okay, these are proteins on the surface of our red blood cells. And the purpose of these red blood cells, these immunoglobulins, okay, if you remember from our cell unit, that we often have proteins and sugars on the outside of our cells that aid in identification and communication, right? So the blood type is the way that your immune system, the white blood cells, will read these immunoglobulins on your red blood cells, and that's how they know that they belong there, right? And there are different types of blood. And so let's go ahead and draw those ones out, okay? There are actually 3 possible alleles. Okay, there are 3 alleles in blood typing. You can have immunoglobulin A, which is represented by capital IA. And you'll see why it's capital in a bit. And what that does is it codes for a protein that we're going to represent with little triangular spikes. Okay. You can have IB, which is another protein. It's a different kind of protein. Okay. And we're going to represent those with little half moons. And finally, we have lowercase I. Now, notice A and B both have an uppercase. They both code for a protein. But when we use lowercase, what are we talking about? We're talking about complete dominance. Okay. And so in complete dominance with this individual, they don't make the spikes. They don't make the half. They make nothing. Right. And so this one is referred to as type A blood. This one here is referred to as type B blood. And then you guys should know, if you don't make any kind of immunoglobulin, then you are type O. And that's actually me, I'm type O. All right, so you have 3 alleles here, but does that mean an individual can have 3 alleles? Absolutely not. Remember, these alleles still sit on a chromosome at a certain gene locus, and every person, because they have 2 homologous chromosomes for a given chromosome number, can only have 2 copies. Okay, if I remember correctly, I think it might be chromosome 7. I might need to look it up and get back to you. I forget exactly what chromosome number our blood type is found on. It's an easy one to look up. Okay, maybe you can look it up and let me know. But the point is, even though there are 3 alleles, everybody still only has 2. Okay, so we want to ask ourselves, what are the combinations that we can come up with of IA, IB, and O, and what does that mean for blood typing? Okay, and so we're actually going to see both codominance and complete dominance, totally opposite ends of the spectrum here, but we're going to see both of them show up in this example. Right? So let's take a look at this. Let's do genotypes here. Let's do, we don't need to do the percentage vote thing. Okay, I'm just going to focus on what proteins we actually make on our blood cell, and then what kind of blood type the phenotype is. Okay, so the phenotype is what is the blood type that you have. Okay, so here we go. Let's start with the easy one. If you are homozygous for A, IA, okay, your cells, your red blood cells here are going to have proteins that are all spiky on the outside, right? Because 100% of the vote is saying, oops, that one's not spiky enough, is saying make A, and so your blood on the outside is going to look like this. Okay. Now, is this the only way to get a blood cell with only all spikes? The answer is no, because we also have complete dominance taking place. So in other words, if I have IA and a O allele, the lowercase I, both of these are going to produce exactly the same kind of red blood cell, right? And again, remember why that is because with complete dominance, I don't want to get tripped up here, the heterozygote, right? Like IA lowercase I is kind of the equivalent of like the big P little p, right? One dominant, one recessive. So if a heterozygote shows up in this situation here, it's going to have the same protein expression as somebody that is homozygous dominant, like this IAIA, right? And the result of this is that this person is going to be referred to as a type A blood type. Okay, so if you're type A, you could have IAIA, you could be dominant, homozygous dominant, or you could be heterozygous. Same thing applies for the Bs. Okay, if you're IB, you're homozygous for B, or you're IB lowercase I, in other words, you're a heterozygote, your red blood cells will look exactly the same because B is completely dominant to O, just as A is completely dominant to O, right? If I didn't make that clear enough, I'm going to do this up here. IA and IB are equal to each other. They're codominant. Oops, I totally wrote that wrong. Sorry. IA and IB are dominant to each other, codominant to each other, but both of them are greater than or dominant to O. Okay, so we see complete dominance in both these situations here. And so the result here is that your red blood cell is going to have those half moon proteins on the outside. And the result is that when you get your blood test back, they're going to say that you have type B blood. Okay. Now remember that your blood type dictates what kind of blood you can receive, right? This is the whole reason why you want to know your blood type, is because if you're type A, you can only receive blood from other type A's, because your white blood cells are used to seeing all these spikes. And so if you're type A and then I'm a doctor and I'm messed up and I give you type B blood, when I inject that B blood and give that to you and it enters your bloodstream, your white blood cells are going to see these half moon proteins. They're going to go, whoa, 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 that doesn't belong. And they're going to cause your blood to clump up and basically kind of turn into jello, right? It thickens so that it can't circulate around the body. And of course, that's really dangerous, because if you're getting a blood transfusion and you pump like a liter of type B blood in your body and it's everywhere and all your blood clumps up, now the blood can't flow, which means you can't deliver oxygen and you can actually die from that. Okay. And that's why it's really, really important to know the blood type. Okay. So A's have to get it from A's, B's have to get it from B's, so that the proteins on the outside match. Okay. Now there's an exception to this. And this is where things get kind of interesting. Okay. If you have IA and IB, so they are heterozygous, right? But both are codominant to each other. So there's no recessive allele here. Well, who wins? Well, remember I told you codominance means that they're equal to each other. And so when we look at the proteins, what we're going to find is that the genetic vote, half is saying make protein A, half is making protein B. And so what they're going to do is they're going to produce both. You're going to see in one cell on the outside both A proteins and B proteins being made. Okay. And what do we call this weird hybrid individual? These individuals are called type AB. Right? Now, type AB is actually kind of a nice thing, because if you're type AB and you get in a car accident and you need blood donated to you, who can you receive blood from? Well, obviously you can get it from other ABs, but you can also get it from type A and you can also get it from type B. Why is that? Because your immune system is used to seeing both kinds of proteins on the surface of your red blood cells. Right? So type AB is referred to as the universal acceptor. They can get blood from anybody as far as the blood types go. Okay. And then finally, kind of sad at the very end, the last option that we have is the homozygous recessive individual. Okay. If you inherit from both your parents the O allele, which is what I have, then your red blood cells have nothing on the outside. Okay, no proteins on the outside. And that's fine, because that's what your immune system is used to, and that's how your immune system knows that those red blood cells belong there. Okay, and these people are called type O. Now, you probably have heard of this before, but type O is not the universal acceptor, they are the universal donor. And the reason for that is because type O's can give their blood to everybody else because there's no proteins to read. And so even though they don't have the same blood type, the immune system is not going to trigger that blood thickening response because there's no proteins to read. Okay. And that's why O is the universal donor. Now, the sad part of this, and this is kind of messed up, right? AB can take blood from anybody. You can get blood from A, from B, or from O. Okay. But ABs can only give to who? Other ABs, right? If ABs give it to type A, their immune system is going to freak out because they're going to see the B proteins. If they give to a B type, they're going to freak out because they're going to see the spiked A proteins. And if AB gives their blood to a type O, their immune system is really going to freak out. They're going to have two proteins that they don't recognize, and it's going to cause that blood to stop flowing. Okay, so AB is a universal acceptor, but it can only give to other ABs. Whereas type O is a universal donor, but who can they receive from? They can only get it from other type O's. Okay, so I don't know if you guys know your blood type, but if you don't, maybe next time you go to the doctor, you can ask. It should be in your chart somewhere. They can do blood tests pretty quickly now, and sometimes they might even do it just as a double check. Like if you, God forbid, you're in a car accident and you lose a lot of blood, they can check your blood pretty quickly and then know which one you're going to get. Okay. Now, a lot of people are probably wondering this during this lecture, well, what about plus and minuses? Okay, like you heard of O positive, O negative. That is actually a separate protein called the RH factor. I'm not going to get too much into that, but that's also inherited in a similar fashion in terms of complete dominance. So you either have it or you don't. Okay, so if you have the protein, you're positive. If you don't have the protein, you're negative. And so the best type of blood to donate is O negative because you lack both the proteins, the immunoglobulins on the outside, and you lack the Rh factor protein, which means you can give that blood to everybody. Okay, the positives it's a little bit trickier. Okay, so here's another opportunity to do some practice. I'm going to give you kind of a complex, I'll give you two. Okay, let me start a new page here. Problem 1 is going to be a simpler one. Okay, let's just do this. If you have a man that is type AB and you cross that with a woman that is type A, okay, and I'll just tell you her parents, one of her parents, let's say her mom is type O, okay, tell me, give me a Punnett square for this situation here. What would their kids end up being, or what are the probabilities of the blood types in their kids? Okay, let's start with this one. It's a little bit simpler. All right, what are their kids? And then if you can tell me anything about this, so the mom was type O, but what was their dad? What could have their dad been? Okay, this lady's dad. Okay, and by the way, guys, I know I'm writing A, B, and A. That's their phenotype here. Okay, so this is when I write AB, this is different from genotype. When we write genotypes, we're going to use the I system. So this individual is IA, IB, and this individual is type A, so she's going to have an IA and something, and that's what I want you to figure out. Okay, so go ahead and pause the video right now and see if you can work it out. What are the kids going to be, and what was this parent up here? Go ahead and pause. Did you do it? All right, let's check out how you did. So let's figure out the mom first. Okay, we got to do that before we figure out the rest of this. And by the way, this is a terrible Punnett square, so I'll redraw it. Also, we'll get rid of that question mark. So if the mom, the woman here is type A, you know that she had to get that A from this parent, right? So this parent has to have a genotype of IA and something. And we don't know what that is, but actually it doesn't matter. It could be a B, it could be an O, or it could be another A, right? But that's where she got the A from. Why do we know that? Because her mother is a type O, which means her mother had a genotype of lowercase I, right? And that means that the other allele that she has to have must be that lowercase I, right? So now that we know that, let's go ahead and plug this into a Punnett square. Okay, I'm making it a little bit bigger just because we have more letters to draw here. So the man is IAIB, right? He's AB. And so one sperm is going to be IA. One sperm is going to be IB. Okay. The woman is going to be on the side. She has an IA as well as a lowercase I. And so hopefully what you did is you fill this out as you would normally a Punnett square. Okay, sperm with sperm, eggs with eggs. Generally, it's good to put the A's first and then the B's first, because then you don't get confused thinking there's a BA blood type. It's the same thing, but we usually put the A's first. And then we have an IA with a lowercase I, and then an IB with a lowercase I. Okay, so we filled that out, and then what do we have? Well, it looks like based on this, what they're going to have is, for their kids, there's a 50% chance that they're going to be type A, right? They could be homozygous A or heterozygous A. There is a 25% chance that their kid is going to be type AB. And finally, there is a 25% chance that their kid will be type B, okay, being a heterozygote. But there's no probability here of them being type O, because both parents would have to have a copy of that allele. Okay, let's do a second one. And for this one, I'm actually going to use some of the questions from the genetics problems. So I'm not officially assigning this, but if you guys go to page 2, let me share a screen here. Here it is. On the genetics problems that you guys did today or last night, the second page are all the blood typing questions. Okay, and let's just go down and let's just pick one of these. Let's do 18. I don't know. I'm just picking this one. Okay, a man with type A marries a woman with type A blood. They have 8 type A children and 1 type O child. What are the genotypes of the father, mother, the 8 type A children, and the 1 type O child? Okay. If I were you, I would go ahead and do a power box here just to kind of clarify everything, because there's a lot going on in here. Okay, but go ahead and pause the video. However you want to work on it, work on this one. Let's see what you end up getting. Okay, let's see how you did. Let me go back to my screen here. So the second problem we're looking at here is a man is type A, the woman is type A. Okay, remember these are phenotypes, not their genotypes. So you have to ask yourself, if they're type A, what are their genotypes? Well, both of them, to be type A, must have at least one IA, right? And something, right? And then what they tell you is that their kids, they have, what is it, 8 kids? 9 kids? They have 8, okay, they have 8 kids plus 1. Okay, so out of this, they have 8 kids that are going to be type A and 1 kid that is type O, right? And so out of these kids, out of these 9 kids, which is the easiest genotype to figure out? It's going to be the O. We know that the O must be homozygous recessive, right? And if they're homozygous recessive, then the parents must be IA lowercase I, both of them. It's the only way you can get that 1 kid, okay? And I know that you're like, but the ratios. Yes, technically, if we did the Punnett Square, you would see that the probability of this is just 25%, right? And 1 out of 9 is not exactly 25%, but that doesn't matter, right? Because the only way to get that O is to have a homozygous recessive individual, okay? So that tells us that this kid is homozygous recessive. Both parents are heterozygous IA lowercase I, and that means all the kids here must have what? Well, there's a couple options here, right? If we do our Punnett Square, what you found was that some of the kids could have been IA, homozygous dominant. And actually 1 out of 4 of them, right, would be IA. And then 2 out of 4 of them could be heterozygous, where they're IA and lowercase I. Okay, so hope that makes sense. You can, I'm not assigning it officially, but that page, that second page, has a lot of practice problems, and some of those can really show up on the test. So you want to make sure that you know how to do those. Okay, I'm going to close with this. I have no idea how long I've been doing this for, but we'll find out at the very end once I upload this and all this. But I want to end with one really interesting case study. Okay, and that is looking at something called Tay Sachs disease. Tay Sachs disease is a neural disorder. Okay, this affects the brain, and the way this works is affects enzymes in the brain. We have this enzyme that has the job of breaking down toxins in the brain. Okay, and if you're able to break down the toxic waste products of the brain with this enzyme and clear it out, then your brain is going to have healthy function. Right now, in individuals that have Tay Sachs disease, they like show symptoms of it. They have symptoms that include what do you call them? Not brain aneurysms, seizures. Okay, and there's just different malfunctions of the brain because the toxins are building up because the enzyme's not working. Okay, so this is an interesting case study because what is happening in terms of genetics and what's happening in terms of the type of inheritance. Okay, well, here's how this works. Okay, if you have, and I'm going to use I'll just use big T, little t for now. Okay, so Tay Sachs disease is a recessive disorder. Okay, if you're homozygous dominant or homozygous recessive, that's pretty easy. Okay, because if you're homozygous dominant, that means you make all the enzymes and you have normal health. Okay, you don't have Tay Sachs. If you're heterozygous, that means you make none of the enzyme. Oh, sorry, let me take that back. If you're heterozygous, you make 100% of the enzyme, but it's a malfunctioning enzyme. Okay, that is, I'm going to put malfunctioned. So it doesn't work, right? And if the enzyme is made, but it's not working, then you're going to have Tay Sachs disease, right? As always, what's interesting is what happens in the heterozygote, okay? Turns out if you're heterozygous, what you're going to do is you're going to make 50% of the enzyme is going to be functioning, and 50% of the enzyme is going to be malfunctioning. And so what happens to this person's health? Well, it turns out that if you have at least 50% of the enzyme working, you don't have Tay Sachs disease. Your brain function is normal. You don't get seizures. You're not at risk for all these neural disorders. Okay. So here's the question, guys. What kind of dominance is this? Is this complete dominance? Is this incomplete dominance? Or is it codominance? Especially if you're in a breakout group right now, go ahead and pause the video and talk about it with your group. Okay, so like a little case study, like with House. What is going on here? What kind of dominance is being used? Okay, pause the video and then we'll come talk about it. All right, so let's break this down. Let's start on the largest level, okay? On an organismal level, on a medical level, like in terms of how the symptoms present. If you're a doctor and this person comes in, do you see a difference in the health of a person between that's heterozygous compared to a homozygous dominant? The answer is no. Both of them present as healthy and normal, right? So in genetics, what we just finished talking about is if the heterozygote has the same phenotype as a homozygous dominant, then what kind of dominance is that? That's going to be complete dominance. Okay, so you could be like, okay, it's done, but we're not done. Here's the reason why. If you look now at the enzyme level, what is actually happening here? Well, you have kind of a blend going on, right? Because you're not it's not that you make 50% of the enzyme and then that's it, right? If that were the case, then it would be like incomplete dominance, right? What you have is kind of this weird hybrid where you're making both enzymes, you're making the functional ones and the malfunctioning ones. And so what does this mean? That means that this one, if you're making both versions of the protein, that's kind of like an example of codominance, right? Because you're making both proteins that are equal to each other. Just like if you're type A and IA, IB, you're making both proteins at the same time. So on an enzyme level, that's codominance, right? But that's not all, because these enzymes are not functioning, right? So what's happening is if you look at it from a waste perspective, what percentage of the waste is actually being cleared out? Only 50% of it, because only 50% of the enzyme is actually working. And so on that level, in terms of the metabolic function of clearing out waste, what we see is a halfway point between a functional enzyme and a non-functional enzyme, which means that's kind of like incomplete dominance, right? So here's the point I'm trying to get at, guys. I told you at the very beginning, genetics is really complicated. It's not just as simple as dominant versus recessive, right? And in the case of Tay Sachs disease, what we find is that depending on which perspective or what level you look at it, you know, whether it's like an organismal level or a molecular level or a cellular level, you could classify this case study as being an example of complete, incomplete, or codominance, right? And I know some of you guys are like, that really bothers you. You're like, I need to know the answer, but that's the whole point I'm trying to make, okay? Is that genetics is complicated, it's not black and white, and there's a lot of in-betweens. And that's why we call this whole thing, going back to my very first slide, where is it? If I can find it, there it is. That's why we call this a spectrum of dominance, because in many, many cases, it's not one or the other side. It could be a little bit of all of them, okay? And so I just want to point that out. That's kind of the whole lesson for today, is for you to understand, number one, what does dominance actually mean? And then what are some other exceptions to complete dominance that we see in the natural world? Okay, so I hope that helps. I know we haven't done this format yet, but I hope you found it helpful. The nice thing is, because this is posted on YouTube, unlike most of my lectures, you can always come back to this and listen to it if you need a little refresher. Okay, and if you have any questions or anything I went over here, if I made a mistake, I might have done that. Feel free to write that down, and then next week when we meet on Monday and Tuesday, we can spend like 5 or 10 minutes at the beginning of class going over this. Okay, so hope that's helpful, and I hope you guys have a great weekend. I'll see you on Monday and Tuesday. Thanks.